Okay, we'll be continuing with the topic of control of gene expression in eukaryotes. We'll be looking at the post-transcriptional level. How is gene expression controlled at this level? Alright, so obviously transcription will have already occurred, which means that at the chromatin level, the chromatin would be loosely coiled with histone acetylation, and that the transcriptional control would have allowed for transcription to occur. Okay, so that's where we can have the post-transcriptional control. We'll be looking at the formation of mature mRNA and how is that process controlled, and also the type of mature mRNA that is produced after post-transcriptional modification via alternative splicing, and how is that controlled. All right. So again, we are not looking at the process of post-transcriptional modifications that has been covered under protein synthesis, right? So we are not looking at the process of protein synthesis. We are not looking at how genes are expressed and how proteins are made. We are looking at the regulation or the control of this process, the control of gene expression, right? So I'm not going to go through the different types of post-transcriptional modifications here. The focus would be on the formation of the mature mRNA, right? How is that controlled, right? So there would be some control in terms of the rate of production. So the rate of post-transcriptional modifications would be controlled, right? And also after the formation of the mature mRNA, it will be transported into the cytoplasm, right, out of the nucleus. So that is also a controlled process. Okay, but then our focus would be more on alternative splicing, right? So alternative splicing is a process where one gene, right, giving rise to one pre-mRNA, that pre-mRNA can be spliced differently to form different mature mRNA, as you have learned before, right? Something like this, right? So this process is a regulated or controlled process. The alternative splicing is not random. Okay, so when we look at alternative splicing, we are looking at different combinations of exons that are spliced together. Okay, so all the introns are excised. I think I have mentioned this before under splicing when we went through protein synthesis. Right, so let me just repeat that. Introns are all excised, and the exons can be spliced together in different combinations. When we say different combinations, what we are saying really is some exons might be included in the final mature mRNA and some may not be. All right. So that's what you see in this example here, right? So the one on the left most, right? It has got all 5 exons included, right? And the other 2 would have missing certain exons. So the one in the middle is missing exon 3. So exon 3 is not included in the final mature mRNA. It is not spliced. Okay, and for the one on the right, it is missing exon 4. Okay, so if you take a look at the sequence of the exons, yeah, because we have put in the sequence, right, 1, 2, 3, 4, 5. You see that the sequence don't get mixed up, right? It will still be sequential. It's just that certain exons would be missing in the final mature mRNA via this alternative splicing. If you think about the process of splicing, how the spliceosome bring the different exons together to form, remember how that happened, right? If you forgot that process, please go back to the protein synthesis notes. Right? So if you think about the process of splicing, you would understand why there won't be the jumbling up of the sequence of exons, right? Exon 1 would always be the first, and then followed by exon 2 and so on, if they are in the final combination of exons in the mature mRNA. Okay, so if you don't understand that, maybe you can give me an email. I will maybe explain to you. All right, but then I thought you should take a look at the splicing process and try to understand it yourself. That would be better for your own learning and understanding. All right, so anyway, what we are saying is the alternative splicing process is regulated, right? So it's not random. So let me give you an example, right? In human cells, right, this early myoblast or early muscle cell, what happened is in the gene coding for muscle production, right, there are 5 exons, right? But 2 of them are denoted 3 alpha 1 and 3 alpha 2. So we have exon 1, 2, alpha 1, 3 alpha 1 and 3 alpha 2, and then exon 4. What happened is there are 2 ways how this pre-mRNA with these 5 exons can be spliced. Right? So you either have exon 3 alpha 1 without 3 alpha 2, or vice versa. Right? So what happened is the first way of splicing would result in the production of a repressor protein, we call our transcriptional control. Right, so this repressor protein would go on to suppress development or differentiation of the cell into these differentiated muscle cells. But the other way of splicing, right, with the 3 alpha 2 would give rise to actually an activator protein, we call transcriptional control again, which would allow for gene expression, which in the end causes differentiation into muscle. So the cell would differentiate into muscle cell. Okay, so you see that these 2 different ways of splicing, alternative splicing, give rise to different proteins with different functions. What happened is this early muscle cell, right, before it received the signal to continue differentiation, it would always splice the pre-mRNA in the first method on the left to give rise to this mature mRNA that produces the repressor protein. But after it has received the signal to continue differentiation, it will then switch the splicing method to give rise to the second mature mRNA on the right that would be translated into an activator protein to induce differentiation. Right? So which way the pre-mRNA is spliced, right, it's actually regulated. It is not random, like the cell wouldn't make both mature mRNA and then somehow produce a repressor protein and also produce the activator protein which cancel each other, right? That would be a waste of resource. Okay, so this I would just want to use this example to show you how this alternative splicing is a regulated process. So in the level of post-transcriptional modification, not only do we control how fast mature mRNA is produced and transported into the cytoplasm, we also control which type of mature mRNA is produced via controlling how splicing occurs in alternative splicing. All right, so that's the main gist for this level of control. Okay.